Welcome, everyone. Let me get the screen share going here. I'm just going to wait just a couple minutes for everyone to trickle in. Okay, let's go ahead and get started. And I realize as I'm looking at my screen right now, it's not September, it's October. Welcome to the October 2022 IACFS ME Virtual Journal Club. I'm Rochelle Jocelyn, joined by my host, Lily Chu, and our text expert, Sarah Moynihan. This event is sponsored by the Institute for Neuroimmune Medicine at Nova Southeastern University in Florida. I'm going to begin just by speaking briefly to our ground rules for anyone who hasn't attended a journal club so far. Briefly, I'll introduce our speaker, and then our speaker will spend about 15 minutes sharing content from their article. And then we'll follow that with a discussion time where we have some questions from Lily and myself, and then we'll open to the audience for discussions with our speaker. And then I'll finish with a brief wrap up, and we'll try to finish within the hour. So today's speaker is Arnaud Germain. He is a scientist at Cornell University, a part of Maureen Hansen's lab in Ithaca, New York, in the USA. And he's going to present on his recent work, Plasma Metabolomics Reveals Disrupted Response and Recovery Following Maximal Exercise in MECFS. And, this article really fits some of my major criteria for a good MECFS publication. It has a large, very well-characterized cohort. It includes a carefully constructed provocation, an exertion challenge, and then measurements using an unbiased approach to generate a very large data set, and then an extensive subgroup analysis to really parse that large data set. So those four criteria for me are really what I'm looking for in an MECFS study, and this paper fits the bill perfectly. It really is a decisive demonstration of metabolic disturbance following exertion, and so we're eager to hear more about it. Arnaud, would you like to go ahead and share your screen?
Yes, and thank you very much for the nice words. All right, so I'm going to be talking about this article that we published beginning of this year, March. This is, as you can see, there are a lot of authors on this paper. So Maureen Hansen is the lab I'm working in, and there are two other principal investigators, Dikoma and Andrew, who are also working. They're part of the center that this cohort is coming from, basically. And in the middle here, you have doctors and people who helped to diagnose and collect the samples that we are analyzing. For this paper, we used a cohort of 105 subjects, and that included 45 controls and 60 patients. And within those within the patients, we had females and males unfortunately, as is the case often, we have less males than females. So most of the results that I'm going to be talking about, even though in the paper we address both of them, it's really the females where we have more statistical power to actually draw conclusions. The samples were sent to Metabolon, and Metabolon is a company that is able to characterize a lot of metabolites out of plasma samples that we sent. In this case, they were able to give us data for 1,157 compounds. We also had them give us samples, names for the compounds that they didn't know what they were. So we have data for 224 compounds that don't have full ID. As Rochelle was explaining, the subjects went through a protocol that was designed to induce post-exertional malaise, and that included a exhaustive exercise that usually lasts between 10-15 minutes on a bicycle, where there are a lot of data that is being collected, including oxygen consumption, things like this. And then blood is taken before and after. And this exercise is done on day 1, and after 24 hours of recovery, it's done again, the same one on day 2. And the blood samples are taken right before and right after exercise on day 1, and same on day 2. And those are the names that I gave them: D1 pre, D1 post, and D2 pre and D2 post. So basically what we're trying to do here is we're really looking at the interplay that is happening between ME/CFS, the exercise, and what is happening in the plasma. And so, as you might have realized, that means that we had 105 subjects and 4 time points for each subject. So that means we have data for 420 samples. And so what I'm going to do, I'm not going to go through the article the way it's written, actually. I'm going to go through the article to try and explain how, in my head, I guess, I try to understand this complex data sets, because it's a lot of, I don't remember the number right now. I can do it. It's 420 times 1,500. So we basically have almost 500,000 data points in this data set. So to try and understand it, you can go at it in very different ways. So one of the first one of the things that I did was do a t-SNE plot. And basically this means that you take those 500 data points and you try and reduce the data set complexity to 1 dot per subjects per time point. So this is what this figure is showing. And what comes out of this figure is basically that each individual you can see is a dot. Well, each time point is a dot, and that each individual is a specific color. And what you can see, for example, if we focus on this one, we have the 4 dots at almost at the same spots on that t-SNE plot. So that means that one subject has, how do we say that, has its own metabolic environment, and that exercise is, when you measure that many compounds in the blood, exercise is not completely changing somebody, but we are all different from each other, as you can see here. What could have been interesting, and it's not the case, unfortunately, but what could have been interesting is if we would have been able to see a clustering of, for example, here in the pink, in the light pink, we have the female patients. We can see that they are throughout the plot here, and the controls are mixed in. So basically, when we look at all those metabolites all at once and try to reduce the complexity to 1 dot, we are not separating patients from controls. And you can see the males are also mixed in, both within the females, and both controls and patients. So this is just the gist of this figure. So basically taking this big data set and trying to reduce it to 1 dot gave us some information, but not information really about exercise or about the disease, just the fact that each person has individuality. So another thing that we can do from a big data set like this is do a pathway analysis, which means that you try to group the metabolites that are measured through a pathway. That means that like metabolism works this way, that you have a metabolite that will be changed to another one, and then another one. So you have this pathway, and so this is what you can look at. And those pathways have names. Some of them are more or less complicated. Here, what I'm showing right now is a pathway analysis in the controls. And basically what I'm looking at is at the effect of exercise. So the effect of this on day 1 and the effect of exercise on day 2, and comparing the two. So within controls, this is just showing for females. Within controls, so what I did is I subtracted the value of the metabolite here at D1 post. I subtracted the value of D1 pre. So basically it's either going to be going up or going down or staying the same. And looking if the change of that metabolite here is the same on day 2. Basically, if the metabolite is changing, is the metabolism of a subject is changing the same way on day 1 and on day 2. So this is what this graph is showing. Here at the bottom, you have the pathway impacts. The more impact it has, it depends on how important that pathway is for us to be, living organisms. And then on here on the left, you have the p-value. And what I put this little drop here. This is the regular threshold that you need to meet for something to be significant. So 1.3, when it's a minus log 10 of the p-value, is this value here. So if you can see in this graph for controls, the difference between the effect of exercise on day 1 and the effect of exercise on day 2, there is no pathway that is significantly different between the 2 days. And I just, in the paper, I just labeled the top ones just so we can see what they are. But the point of this is that there is not that many changes. And that means that the subjects are repeating the changes that are happening on day 1 are also happening on day 2 in the same manner. Now, if we look at the same analysis, but this time on the patients, so on the IACFS cohort, you can see that the scale here on the left has changed, and the cutoff, the statistical cutoff, I know it's arbitrary, it's just math, but the cutoff is here, and you can see that there are a lot of pathways that are significantly differently between day 1 and day 2. And actually, I think it's 14 of them. And we discussed a little bit of them in the paper. It's kind of difficult to pick and choose which one you want to talk about without being biased. So when we wrote the paper, we tried to stay more general. I know it can be frustrating, but at the same time, we didn't want to pick what's our favorite, because we don't know which one is really the most important in those. They all look important. So you have a lot of amino acid pathways here. Here, the Warburg effect is something that's talked a little bit in the field of IACFS, and it has to do with oxygen consumption within the cells. And then you have a little bit of fatty acids, which is also something that's in the field. So as you can see, it's difficult to just pick and choose. So we didn't, we just showed the result. Another way of looking at, oh, sorry, I forgot about this. The other thing that was actually interesting, to me, actually, the most interesting is looking at this, is doing the difference of the metabolites 24 hours after the exercise compared to right after the exercise here. And when you do this, so this time I'm comparing controls to patients. So I'm looking at how is the recovery process different between control and patients? And you can see here there are even more. I don't remember the number now. Maybe it's 19 pathways that are above the threshold here, and there are a lot of pathways that are changing very differently between control and patients during the recovery time. Again, we have a lot of amino acids. Again, the Warburg effect is in there. So there is a big, this just shows that there is a huge difference between control and patients. So the controls are able to just reproduce the same, the exercise on the 2 days without having that differences, whereas the patients are not. And then the recovery is very different between control and patients. So another way of looking at this data, again, and try to simplify it, is to do a cluster analysis. That means that you're just going through the what the compounds are looking like, their chemical structure, and then you see what is significantly changing when you group them by that criteria. It is different from the pathway analysis because you could have chemicals that are part of several different pathways, but you will group them together because their chemical structure has been deemed to be close enough by chemists. Like, this is just a pure, like, no biology per se. It's, well, biology comes after, but like the classification before is not, there is no biology. So I'm going to show 2 figures. This is the female one. And here, so I hope you understood what I explained for the pathway analysis. Here again, I am looking at the delta on day 1 of the controls, how this is, what is changing during this exercise on day 1, then the recovery period, and then what is happening during the exercise on day 2. And I did this for the controls on the left and the patients in the right. And what is interesting here is that you have not many changes happening in the controls during the exercise, or that I should say, there are changes obviously, but I'm only showing, this graph is only showing what is considered significant. So the significant changes are, there are 3 clusters here. Then during the recovery period, you have some that are changing. And then during the exercise on day 2, you have changes again. And what's interesting to see is that, I should also say for the patients, you can see there are more changes during the exercise here. A lot is happening during the recovery in the patients. Many different clusters are significantly different. I need to mention also that the colors, it means that the compounds are going down. When it's blue, when it's red, the compounds are going up. And then when it's purple, some compounds are going down and some compounds are going up within the cluster. And so what I'm not going to go into the names. To me, what's interesting here is you can see that some pathways, for example, the arachidonic acid cluster, I should say, is going down during the exercise on day 1, going back up during the recovery, and going down during exercise on day 2. And this is also happening in the patients. Here you can see it's significantly changing, and it's changing the same way in the controls and the patients. So to me, that's interesting because at the same time, you can see all those other clusters that are not changing in the same way between control and patients. So like there is a big difference in metabolism. So those are results for the females. And we also have results for the males. And what was very surprising to me when I got the results was that you can see that it's the opposite as the females. There is almost no changes happening in the males throughout the whole protocol. The arachidonic acid is still here, behaving the same way as in the females, but the rest is the clusters are not behaving the same way at all, and there is a lot happening in the controls. So interpretation, there are 2 things here that I need to say is that, again, we don't have a lot of subjects, so we have to be careful. Like, this could change completely if you doubled it or tripled the size of this cohort. And then the other thing, I forgot what I was going to say about the other thing. All right, and so I'm going to finish with the so the last thing, I know this is the first thing in the paper, but the last thing that I think it flows better that way when you think about the data set that way is now when you do statistical analysis, that means that I'm really looking at compound per compound, which one are significantly different. And here, I'm just going to show this part of what is happening. Those are the 4 different time points. This is when you mix females and males. This is just the females. This is just the males. And the threshold here is a p-value that I'm showing. And this is just counting the metabolites that are significantly different between control and patients. And you can see here in the females, you have a lot of metabolites that are significantly different between control and patients at the 4 time points. Exercise is increasing the differences between control and patients throughout the protocols, throughout the 4 time points. And what was interesting was also that for the males, there is exercise on day 1 or on day 2 is not increasing the number of changes. It's really during the recovery period that the differences are doubling. But the exercise by itself is not changing, whereas in female, it is changing. It is increasing it. When you mix them together, you can see that it's not really a good way to analyze the data when you know that there are differences between females and males, but we still showed it. And here at the bottom, I have the unknowns, and you can see that the unknowns are making quite a good portion of the metabolites that are changing. I think am I out of time? 1 minute? Yeah. I have 2 more slides, but I'm just going to say this one thing, is that there is no metabolite that is the metabolite that we found to be the difference between control and patients. And even when we have metabolites here, this is the metabolite that is the most different at the first time point at baseline, before any exercise. Here I'm showing this is total control in patients, female control in patients, and male control in patients. You can see that even though statistically it's highly significantly different, you can see there is a lot of overlap between the subject, between control here and patients. So like there is no like, oh look, there's this metabolite that is brand new that is just appearing in controls and it's not in patients, and this is what we need to treat. That paper, we didn't find that. We just found a lot of differences that are happening, the way that the changes are happening. For example, here, just one example is going up in controls. It's going up during exercise on day 1, then going down during recovery and going back up during exercise on day 2. Whereas in patients, you can see that it's not there. The differences are not that big. As big as they are for the control. So again, this paper has, this is my conclusion, this paper has such a big data set. We tried to analyze it as many ways as we could, and then we published it with the data set to let other people go in and like maybe have a better idea than us to look at this data set. Like there is a lot in here that can be used for many other things, including just people who are interested in exercise in controls. So like this data set, I think is going to be very valuable for people who want to take on doing an analysis a different way, or just focusing on one thing just because they have a hypothesis that they want to test, and without having to have subjects go through that very difficult protocol, and they can just use the data sets and to test it, and then maybe if it's positive, then they can keep working on it. So I'm going to stop here, and I'm ready to try and answer all the questions.
Thank you. I wonder if you could actually back up a slide, and we'll leave that final data set available just while we discuss for something to look at there. So I want to ask, you mentioned that this data set is available for others to use to study exercise, but that's with the caveat that the control population is fairly sedentary. I think could you speak a little bit to the activity matching attempt that was made between the MECFS and the control population, and the role that deconditioning might play in this data set potentially? And I'm curious, kind of as a third part to that question, whether there are pathways that are known in deconditioning that you can look in these data as an internal control to parse kind of how deconditioned or relatively, if there's a deconditioning difference between the controls and MECFS population.
Mm-hmm. Good question. So Okay, I'm going to try and answer all of them. I don't know if it will be in the right order, but so I'm thinking about the exercise right now. So what's interesting is usually studies that are done on exercise are done on males that usually are athletes. So there is almost no paper that has studied sedentary females doing exercise. So in that manner, it is an interesting data set to look at for other people. Obviously, it's not enough subjects. They would need to do more, but it's a good start. But yeah, it was interesting when I was going through the literature, like every time it's, young athletes and they're like, look what the effect of exercise is. And it's mostly males because I don't know. Well, I'm sure we all know. So that's one part of the answer. So the way that the project was written was to actually try and match the activity level of the subjects between the controls and the patients. That was a decision that was made by Maureen, by my boss. I think it's a good one. Before that was not taken into consideration, at least not in our lab. So I think it's good to try and take that into consideration. Concerning the deconditioning, I don't think I know, to be honest. I can't answer that question. I don't know if
It's a question that comes up in MECFS research frequently, and I wondered if this data set had one metric in there similar to the arachidonic acid. You see that pattern happening consistently across both populations, and we don't know whether arachidonic acid metabolism is modulated, across differing deconditioning states. But something like that might be a known function of a deconditioned individual that could be quantitatively compared between the two populations would be useful for other studies. If that could be extracted from these data, it could be a useful method of developing activity matched cohorts, I guess is my suggestion.
I see.
Yeah. So it seems to me that data might be accessible in this data set.
Yeah, okay. I was more thinking that your question was not necessarily, well, negative. I don't know what the right word would be, but like more in a way that says like, it's so what do you think? Do you think it would be better? Which would be better, like to have the activity matching or not?
Definitely, activity matching is the best you can do to control for a confounder, right? Is the best attempt, and that's what I was wondering, if within this data it could be a more quantitative control and more biological measure of controlling that other study groups could use. Just a kind of a curiosity.
Ideally, we would have like people that are not athletes, but just people that are active. I don't know, maybe walking 1 hour a day, or like running half an hour a day or something. Then the sedentary controls, and then ideally you would have 3, and then maybe I could answer your question about, are there pathways or something that we can figure out, like a quantitative measure of the actual activity. So the way the controls were screened for activity was questions that were asked to them, but also a part of the cohorts. Actually, I don't know if it was done on the controls as well. I think so. Maybe just on the patients. Anyway, I think it was on the controls. Part of the cohorts, 2/3 of the cohort actually wore bracelets, monetary bracelets. I think 10 days or 5 days before, and then 10 days after, or something like that. This is data that is not in our hands because we're part of a center, and it's being analyzed right now, and it will be made available as soon as they're done. But so all of this is looked at, and maybe once we have more pieces to the puzzle, that question can be looked at again to see if we can find something.
Yeah, thank you. Lily, do you want to ask a question?
Sure. So hi, everyone. Arnaud, that was an excellent presentation. I just thought your group has done so much work on this. And those figures are really beautiful. And I'm glad you clarified, because I had some points about them I had questions about. One question I had was, we all know that the onset and duration of PM, much less the intensity and the symptoms, can vary a lot between individuals. So I was wondering if you had gathered any symptom data, and whether at least people could say, when I had my blood drawn, I was already in PM, or whether they said, my PM was decreasing by the time this blood was drawn, or something like that. Any thoughts about that?
So we had so as I mentioned, hold on. As I mentioned, we have some people that wore bracelets. We also had people answer. So we had people and subjects answer many different surveys: the SF-36 and the Bell scale and the SSS. I never remember what it stands for. It's like symptom severity scale, I think, actually. And so we have a lot of data about the symptoms, and those have been answered after the exercise on date. So I think throughout 10 days as well, every 2 days, they would rate how the symptoms were evolving. Are they getting better? Are they getting worse? When is the peak, I guess, of PM? And then are they, after 10 days, like, are they symptoms receding a little bit, or they're still raising, things like that. So we have all this data. It was not, well, two things. I didn't have the skills at the time to use them. And anyway, they were not all available at the time, like definitively available. So it's also in the plans of being looked at once we have everything, I guess. We have a new colleague, a new postdoc in the lab who's very good. Catherine Glass, she's very good at looking at things like this, like correlations. And so we will be revisiting that. It needs to be done. Yeah, it was not done at the time, but we are still.
I mean, it's a lot of data, so I can understand this is very complex analysis. But I was just thinking, if at least you could figure out, this person had PM at this time, or they were at their peak, it might help clarify some of the points, in the data. I also have another question, but I'll hold off for a minute.
I just wanted to finish commenting. That's a very good point, because if right now I'm considering everybody is reacting the same way to the exercise in this analysis, and if you could find out that a subject got PM, I don't know, when they were doing the exercise on the second day already, or if the PM arrived 6 days after this complete protocol, then you could statistically correct for that, and maybe it would show, like, that metabolite had risen a lot in the person who got the PM right away, but it was rising. Even though we don't have the actual data, you can statistically anticipate that it might have rose throughout the 6 days after. And so, yeah, that will be done. We're thinking about it, and it's very important.
I have a similar question about clinical variables. Did you perform any, I know you did, PCA and you did some unbiased approach analysis. Did you manually perform stratification across any other clinical variables besides sex? So, things like disease severity or disease duration, or various symptom constellations, kind of manually within the MECFS cohort?
Like I said, I didn't have the skills at the time to do it that way, like to do it in a rigorous way, I should say. The way I did it is for the most interesting metabolites that came up as significantly different, I colored, or I looked at where they were, like manually, basically, like where if there was anything happening, if there was some sort of a gradient going from like these symptoms to more symptoms, or like I looked at 4 or 5, I think, like the Bell scales, things like that, try to see if it fits. You know, the more you have of that metabolites, the worse you are on the scale, things like that. I did not see anything, but that was really anecdotal, I have to admit. So I can't say it's been done, or it's not been done properly. And by luck, I did not find anything, I should say. You're muted, Rochelle.
Sorry. I'm curious if you have any in vitro experiments planned as a follow-up to some of the pathways that you did discover. So I think in your paper you mentioned the malate aspartate shuttle and glucose alanine cycle as some of the energy production pathways that were notably impaired. Are there, I guess, does the Hansen lab in general have plans to follow up on some of those implicated pathways in vitro?
So
or ex vivo, potentially with a provocation, I should say.
So when you say in vitro, you mean cell cultures?
Or ex vivo PBMC analyses of those pathways.
Yeah. So that's one of the things that's important to remember is this data set is coming from blood that doesn't have cells in it. So the red blood cells have been taken out and the PBMCs, sorry, I should say it's coming from plasma, the plasma part of the blood. So it's mostly cell-free. So the result that we are measuring, it's I want to say dangerous, or it's risky to try and extrapolate to where those changes are coming from. It's important to remember that the blood is literally like all over our body, going through like muscles and organs, and so and in there are the immune cells, of course. So we are working in the lab on PBM on immune cells. There is grad student Jessica Maya, who's, I'm sure you've heard her talk places. Like we are working on those cells and their metabolism and their activity, but we don't have plans to test in vitro right now. We don't have plans to test in vitro because I don't know how we would test it, to be honest. Like,
right.
I haven't thought of, we haven't discussed of any hypothesis that we could test directly, and what cells we should be using for that. I mean, right. Like, should we be using muscle cells, like, or immune cells? Like, yeah, you can do a lot of experiments in vitro. It's not really, yeah. I
think that was my question was, did any of the pathways that emerge in this large data set send you in a specific direction that could be tested in a dish? But it wasn't immediately clear to me either. So that's the nature of the question. Yeah.
The short answer is, I mean, we always try to think of what we could do after following that. That's not currently what we're thinking about. I should say, and you've heard many of our talks, you can see that we are currently we're using those samples and analyzing them in many, many different ways: metabolomics, proteomics, extracellular vesicles. So like right now, I would say it's more like we're looking at the samples as many ways as possible, and then once we have published all this, maybe us or somebody else will be able to say, hey, this is what looks like it's interesting coming out of this. I personally believe it's going to be very important to, and my lab also believes it's going to be very important to do a multi-omics analysis of all of those data sets. Like, what is, when you have this change in metabolites, what is happening at the protein level? Is it matching, and is it going the same way? And then maybe go all the way to genes and things like that. And so, like, I think it will. Yeah, I was going to say it's not going to be faster, do it that way, but it's going to be more thorough.
Mm-hmm.
And ideally, at the same time, you would do maybe some testing like you mentioned, but that's not really our specialty in the lab.
And is that something that your lab captured alongside with all of those clinical data and the metabolite data? You also have like RNA-seq and protein analysis in parallel?
Yep.
Yeah. Okay. So that's a really, really large data set.
Oh yeah. I think I must have presented, or Maureen presented it at IACFS. No, I don't remember. But anyway, we have a lot of data that we are analyzing right now, and we're not looking to have more experiments to do right now, but it will come very soon to move forward. But right now, it was a lot of work to collect all those samples, especially during the pandemic and everything. So it's like there is a lot in here that deserves to be analyzed, with all the efforts that's been put by subjects to like do as many testing, as many experiments on those blood samples as possible.
Yeah. Lily, I think you had additional questions. I also want to suggest to the audience, if you have questions you'd like to ask Arnaud, please just use the raise hand feature and we'll unmute you. Did you have something else, Lily, you want to ask?
Yes. So anyone, please raise your hand and we'll call on you. One question I had was trying to make sense of the huge amounts of data that you have. And it's been like over 2 decades since I learned about human metabolism. And I was looking at, I'm not that familiar with them, but I know there are people working on what they call in silico or e-models of human metabolism, where they have a program that puts together all the data of known metabolic pathways in people. And so I was trying to look at some of those this week, and I was wondering if anyone in the group has looked at them, where you can put in like, here are the metabolites that have changed, or the metabolites I'm interested in, and they can highlight within the huge metabolic map where those pathways are. And I even wondered if there were any models where you could manipulate it and say, okay, this pathway is out, what would we see in people? So I was wondering if your group had any opinions about that.
Another good question. So I am not an expert in that. I have played a little bit with the software that's from QIAGEN. It's called IPA, Ingenuity Pathway Analysis. I have fed in there the data sets, and then played a little bit to see if it came back to be similar to some other disease, or like behaving the same way as other things, like that is in their database, their library, I should say. And I didn't find anything interesting. A lot of times you have, it's difficult, because I think those have to be biased when you do that, because a lot of the research that is happening is, for example, on cancer and things like that. So there are way more data sets in the libraries that are about cancer. So then when you find when there is some similarity, it just tells you, like, oh, it's very close to whatever cancer. And you're like, well, no, like you have to dismiss that because, well, it's not. So, when I used that software, nothing popped up. But again, we will be looking at that. I think once we have more data sets, like the proteins, for example, in combination with the metabolites, and we have it already, but just like put them together and try to analyze it that way, it might give us some more clues or insight about your question, like how is it similar to other things happening, like to other diseases that are known and have been more studied, or things like that. Yeah.
Yes, the thought is that if those e-models are helpful, then they might be able to not only help figure out the pathophysiology of disease, by manipulating known knowledge or other known data sets, but also for figuring out what could be repurposed as a drug, or even testing new treatments without necessarily having to test them in humans or in animals. And so this in silico analysis is something the drug companies are using to some degree a little more in the past, but it's still very, it's very preliminary, very new.
Yeah, so the software I was mentioning has also a section about drugs, like it is included in there. But I don't think I'm qualified to look at that because I don't know. Like, I think it would be more useful for somebody else somebody who's knowledgeable about drugs to be looking at that in terms of pathways and drugs, how they're affecting, and then be like, hey, we should like focus, we should dig a little bit deeper on that one, for example. So like basically what I'm trying to say is like those are all very different fields every time, and I think it's more it's better used for somebody who is already an expert in that field to actually take that on. And like we are always open. I know there are not a lot of people, but on the call, but we're always open to collaborations, or like once it's published, it's literally like, please take the data set and prove that I didn't do something correctly and find something in there. What I found might not be it, and I won't be mad if somebody else is doing a different approach and find something. So I think it's something that's kind of missing in general in science. There are very few people in the world that go back to published data sets, and it's difficult also because you don't know exactly how everything was done, but that go back to a data set and just say, hey, I don't have to do experiments, I'm just going to just try and analyze all those data sets that are already published. It's done a lot more in RNA analysis data, but not much in metabolomics or proteomics.
It's very common in public health that people will share, and there are even some public databases that people do work on and use to looking for new findings. If that is a possibility, who would people reach out to? Would they contact you or Dr. Hansen for data?
Either anybody in the lab is available, but Maureen Hansen is usually the corresponding author, so she always entertains any ideas that's emailed to her about collaborations, or she just passes it on if it's just an idea or anything. We're always available to be contacted with any question. Even when you read a paper and don't understand a figure, feel free to email me and be like, do you mind writing a paragraph to explain that figure? Or get on a call.
That's great.
I think it's most scientists, to be honest. It's not just, well, I know some scientists are very, very busy, but I think you can always reach somebody in the lab to get more explanations.
Yeah, I think there will be people who watch these videos after this live meeting, and so that information will be very helpful if they're interested in collaborating or looking at some of the data.
Yeah, I think it's important because somebody cannot know everything, and it's better to bring in new ideas from another brain and who has different expertise and ask the question differently, and you can be like, why didn't I think of that?
So audience, if you have any questions, please feel free to raise your hands.
While we're waiting for others, I have a question about the males. So what's up with the males? If you take a random sample of your female cohort, the size of the male cohort, do you see similar results? Have you attempted that?
No, I haven't attempted that. I don't. The problem is because of so when the study was designed, the goal was to not ignore males, which is often done in the IACFS studies. So the goal was to have males just not because we were going to find differences, just to include males. And when we found out that we had to separate them, it's actually not necessarily a good thing for statistics, because when you have 15 and 15 with that many metabolites measured at the same time, you don't have a lot of power for your analysis. It's kind of, which is why I put a lot of the male data in supplemental, because you have to be careful the kind of conclusion you make, because if you add, it could be, you just add 3 more subjects and it will completely change the statistics.
Yeah.
So
that's really my question. Is there any way to get at whether this is a sample size issue, or if there's something really intrinsically different about the male response to exercise, and or the diagnostic process, even thinking upstream, and you may not have a lot of insight into the clinical data that's associated with the males, is there some potentially comorbidity or overlapping condition? Is their diagnosis really clean and crisp, or were there exclusionary criteria involved in the process that didn't sufficiently capture males with other disorders that could be included in this, and confounding, with a small number, just a couple of people who have, some other sort of pituitary adenoma or something, it presents somewhat similarly to MECFS, but really isn't the same disease at a metabolic level, could distort the data. I wonder if maybe a retrospective analysis of their diagnostic process for these patients might help you identify a few that could be excluded.
That's an interesting thought. I can't answer it. We completely I've never met the patients, any of the subjects. We completely rely on the doctors who are specialists. They're the authors on the paper, who are specialists in diagnosis, and then I don't even know, because of privacy. I don't think we would be able to have access to any that you're mentioning. So
sure.
I don't know if there would be a way to get to go the other way around, where we actually ask. We have to go back to the. I don't know how that would. Mm-hmm. That's an interesting idea. I will mention it to.
It's something to revisit. I wonder if a more stringent exclusionary criteria applied to the males might clean it up a little bit. But it's still, with 15, you're reducing that sample even smaller, given what you state as the kind of high individual variability of metabolic function to begin with. You really need an extremely large cohort to make sense of all that noise. But I wonder if it's what jumped out at me at first. First blush was, I wonder if some of these males don't have MECFS, if they've got something else going on, some heart condition, or there's a variety of other conditions that could be excluded.
Well,
that
present similar. I'm not familiar, because I didn't do it, with the exclusion criteria, but they were the same for the males and the females, for sure. They were done by the same people, and I don't think I don't think it is a concern. I don't know, but I'm pretty sure it's not a concern. So, but that's all I can say.
Well, and I'll reiterate my praise of, the well-characterized patients being one attribute of this study, and that the process includes really thorough clinical data capture and vetting with experts. So,
I can speak to that. That's we have experts in the field who we worked with to gather this cohort. So I'm confident that the work was done correctly, as far as it's possible, without an actual diagnostic test.
It looks like we do have a question from the audience. I see Mark Sozinski. Would you like to unmute yourself?
Yeah, for the sake of time, I'll keep this simple, and great job. Love it. I'll definitely be emailing you on that. Were the pre-samples fasted?
No.
So I think right there, that's some variability in metabolite levels, right?
Yes, definitely.
But then you don't want them to do a max test on fasted. So I get it in that population, but that's another interesting question, perhaps.
Definitely. It's there is a lot of limits that come in when you start dealing with human subjects in terms of approvals by universities and things like that. So what you can do with human subjects, and same with animal, but I guess that's stringent. And we did not control for the food that they had before they came. The exercise was most of the time, I believe, done earlier in the day. But there are always things that can happen, and then it's pushed back. And ideally, I don't know if you would want to fast MECFS patients before asking them to do a exhaustive exercise.
Exactly. Yeah.
So, but ideally, I would say you would want to be providing them with the food that they are going to eat before, so that everybody,
right,
and hoping that they eat all of it, but everybody eats the same food, so that you control a little bit for that. I mean, even more ideally, you would have maybe, I don't know, maybe a few days or a week of everybody eating the same thing, but you see that you're starting to get into some complex protocol that is difficult to do apart from being like in setting of a hospital maybe, or something like this, which then you start getting very high costs. Here we definitely didn't have the funds to do that. Unfortunately, I think a lot of time it comes down to there is just no money to, I don't know, buy food for people to eat before, things like that. It's like, because all the other costs that are happening. It's unfortunate, but yeah. So yeah, they wouldn't be tested.
I had a similar question about controlling for diet and hydration in the 24-hour interval between, just with the presumption that the control population would not maybe consider their diet and hydration, with the same level of scrutiny that a patient might. And similarly with any supportive measures, I don't know if any of the patients following the exertion had IV saline for rehydration, but that's one therapy that's really quite effective generally in the population, and could potentially, how much hydration influences metabolic function, but I'm sure it does have some influence. The patient population might hydrate a lot more.
Maybe, but definitely not between the two exercises. There was no like IV hydration for the patients. That's for sure. Like, and to be honest, I think patients for this protocol were more compliant to what was asked to do in between compared to controls. They have more at stake, and they are more. Anyway.
Yeah, absolutely. So we're coming up on the hour. If anyone else in the audience would like to ask a question, go ahead and raise your hand. Or Lily, if you have anything else to add, jump in.
Like I said, also feel free to email me.
Absolutely.
Me or Dr. Hansen.
Okay. Well, thank you very much for your time, Arnaud. We appreciate you joining us here for the hour. And if you want to stop your screen share, I will go ahead and mine up. Okay, there we go. So our next virtual journal club is November 16th, and we'll be hosting Dr. Martina Seifert following her publication, Serum of Post-COVID-19 Syndrome Patients, past patients with or without MECFS differentially affects endothelial cell function in vitro. So it's a serum transfer study in long COVID. Specifically looking at those with and without MECFS. That's been really interesting. So we hope you'll join us for that on November 16th. And then if anyone else has any publications or speakers that they'd like to hear from, feel free to use the Google form that will pop up when you exit this call, and send us a message about what you'd like to hear from in a future meeting. All right, that's all for today. Thank you for joining us.
Thank you very much. Have a good weekend.
Thanks.
Thank you, Arnaud. Thank you for everyone for joining us. hope to see you in November.
Bye-bye.